In this chapter, we will explore various aspects of prokaryotes, including their habitats, relationships, and microbiomes. We will delve into different groups such as proteobacteria, non-proteobacteria, gram-negative bacteria, phototropic bacteria, gram-positive bacteria, deeply branching bacteria, and archaea. The study of prokaryotes has a long history with significant advancement occurring over the years. A notable moment came in 1966 when scientist Thomas Brock discovered that certain bacteria could thrive in boiling water, igniting curiosity about their potential to inhabit other extreme environments, including the depths of ocean, high altitudes, volcanic regions, and even extraterrestrial locations. Prokaryotes play a crucial role in shaping and sustaining our biosphere. They are involved in essential processes that contribute to the health of ecosystems. For instance, some prokaryotes are capable of producing proteins and substances that are valuable in molecular biology, medicine, and industry. One remarkable example is the bacteria Shewanella, which resides in a deep sea environment with limited oxygen. It has developed long appendages equipped with sensors to locate scarce oxygen and possess the ability to digest toxic waste while generating electricity. The diversity of prokaryotes is astounding, as they contribute significantly to the planet's oxygen production and nutrient cycling. Certain species produced more oxygen than the entire Amazon rainforest, while others provide essential nitrogen compounds for plants, animals, and humans. Additionally, prokaryotes inhabit the human body, offering protection against harmful microorganisms and producing vital substances necessary for our health. This chapter will examine the structure, function, and incredible diversity of prokaryotes, highlighting their importance in both natural and human-made systems. In this section, students will be able to identify and describe unique examples of prokaryotes found in various habitats on Earth. They will also learn to identify and describe different types of symbiotic relationships, as well as compare normal, commensal, and resident microbiota to transient microbiota. Additionally, students will gain an understanding of how prokaryotes are classified. All living organisms are classified into three domains of life: archaea, bacteria, and eukarya. This chapter will focus specifically on the domains of archaea and bacteria, both of which consist of unicellular prokaryotic organisms. Unlike eukaryotes, prokaryotes lack nuclei and other membrane-bound organelles, which distinguishes them from more complex life forms. Prokaryotes are found everywhere on Earth, including extreme environments such as hot springs, the Antarctic ice shield, and deep underwater, where pressure is immense. An example of the bacteria, Paracoccus denitrificans, which can survive even when removed from its natural habitat, which is soil. Scientists have subjected this bacterium to gravity forces similar to those on Jupiter using a centrifuge, demonstrating its remarkable resilience. Prokaryotes in human body. Prokaryotes, particularly bacteria, are abundant both on and inside the human body. According to a report, the National Institute of Health, bacteria outnumber human cells by a ratio approximately 10:1. More recent studies suggest this ratio may be closer to 1:1, but still indicates a vast number of bacteria residing within us. These microorganisms thrive in various parts of the body, including the mouth, nasal cavity, throat, ears, gastrointestinal tract, and vagina. Healthy human skin also hosts large colonies of bacteria, particularly in the moist areas like armpits and the navel, though dry regions are not devoid of these microorganisms. Ecological importance of prokaryotes. Prokaryotes play a crucial role in maintaining the stability and health of ecosystems. They are essential for soil formation and stabilization, helping to break down organic matter and develop biofilms. A single gram of soil can contain up to 10 billion microorganisms, primarily prokaryote, representing around 1,000 different species. Many bacteria utilize nutrients released from plant roots, such as acids and carbohydrates, and in turn, they metabolize these substances and return byproducts to the soil, enhancing its fertility. In unique ecosystems like the Dead Sea, salt-loving halobacteria decompose dead brine shrimp and support the growth of young shrimp and flies through their metabolic byproducts. Resilience and adaptability of prokaryotes. Prokaryotic microorganisms are not only found in soil and water, but are also abundant in the air, even at high altitudes. The diversity of bacteria in the air can reach up to 2,000 different kinds, similar to the variety found in soil. Their widespread presence is due to their exceptional resilience and adaptability. Prokaryotes can often switch between different energy sources based on availability, exhibiting metabolic flexibility. For instance, certain cyanobacteria can alternate between conventional lipid metabolism and generating biofuels like fatty acids. Groundwater bacteria can store high-energy carbohydrates when in pure groundwater, but will metabolize these when phosphates are present. Additionally, some bacteria can reduce sulfates to sulfides, but can adapt their metabolic pathways to produce acids and free hydrogen ions when needed. Prokaryotes play essential role in maintaining life on Earth by capturing and recycling vital elements such as carbon and nitrogen. Animals depend on organic carbon for growth. However, unlike prokaryotes, they cannot utilize inorganic carbon sources like carbon dioxide. Consequently, animals rely on prokaryotes to convert carbon dioxide into organic carbon products through a process known as carbon fixation. This conversion is crucial for the survival and development of various organisms. In addition to carbon fixation, prokaryotes are also critical for nitrogen fixation, which transforms atmospheric nitrogen into ammonia. This ammonia is an important compound that some plants use to create various biomolecules necessary for their survival. For instance, bacteria from the genus Rhizobium are known to be nitrogen-fixing bacteria that associate with the roots of legume plants such as clover, alfalfa, and peas. The ammonia produced by the rhizobium supports the growth of these plants by enabling them to synthesize essential nucleic acid building blocks. These plants may then be consumed by animals, promoting their growth and survival, as they may decompose, enriching the soil with nitrogen fixation products that benefit other plants. Another significant benefit of prokaryotes is their ability to assist in environmental cleanup. Researchers have recently investigated the diversity and functions of prokaryotes in human-made environments, discovering that some bacteria play a crucial role in breaking down toxic chemicals that pollute water and soil. This biodegradation process is vital for restoring the health of ecosystems affected by human activities. Despite their many beneficial roles, some prokaryotes can pose risk to human health, as pathogens can cause illness or infections. Additionally, certain bacteria may contaminate food, leading to spoilage or foodborne illnesses, raising concerns about food preparation and safety. While it is estimated that less than 1% of the prokaryotes are human pathogen, these species are responsible for a significant number of diseases. Beyond direct impacts on Earth, prokaryotes also influence humans indirectly, particularly in the context of climate change. The thawing of permafrost in polar regions releases carbon previously trapped in the soil, which is metabolized by prokaryotes. This process results in the emission of substantial amount of carbon dioxide and methane, contributing to the greenhouse effect and climate change. Prokaryotic microorganisms such as bacteria can form unique associations with plants and animals, leading to various interactions. For instance, some bacteria live on plant roots or leaves, obtaining nutrients while providing protective substances against pathogens. Conversely, certain bacteria can act as plant pathogens, employing similar infection mechanisms as those affecting animals and humans. These prokaryotes exist within communities, which are groups of interacting populations of organisms. A population consists of individuals from same biological species in a specific geographic area. Interaction within these populations can be cooperative, benefiting both, or competitive, where one population seeks to outcompete another for resources. The field studying these interactions is known as microbial ecology. Interactions between different species in a community are referred to as symbiosis. These interactions vary in nature, ranging from harmful to beneficial or neutral. Key types of symbiotic relationships among prokaryotes include mutualism, parasitism-predation, amensalism, competition, commensalism, mutualism, and neutralism. Mutualism occurs when both species benefit from their relationships. An example is the mutualistic association between the humans and the bacterium Bacteroides theta iota micron, which aids in digesting complex plant materials. Another beneficial relationship is with the certain strains of E. coli, which provides essential vitamins for humans such as vitamin K. Amensalism is characterized by one population harming another while remaining unaffected itself. Some bacteria produce substances that can kill competing bacteria such as Staphylococcus epidermidis, Propionibacterium acnes, which produce antimicrobial compounds that target potential pathogens without harming themselves. In commensalism, one organism benefits while the other remains unaffected. For example, Staphylococcus epidermidis thrives on the dead cells of human skin without causing harm. This relationship illustrates that classification of symbiosis can overlap as both parties may gain advantages under certain conditions. Neutralism occurs when neither organism is affected. An example includes the coexistence of active bacteria and dormant endospores such as Bacillus anthracis. This form of symbiosis allows both bacteria to thrive in varying environmental conditions. Parasitism describes relationships where one organism benefits at the expense of another, as seen with many pathogenic prokaryotes. Diseases like tetanus, tuberculosis, leprosy arise from bacterial invasions that produce harmful substances in humans. Predation is also observed in prokaryotes. Some bacteria use a range of strategies to prey on neighboring bacteria, including secretion of lytic enzymes, direct contact feeding using cytoskeletal fangs. Some examples are Vampirovibrio, species that feeds on eukaryotic chlorella cells, and Vampirococcus, which sucks the cytoplasm out of Chromatium bacteria. Delovibrio bacteriovorus is a predatory bacterium that kills and eats other gram-negative bacteria. It uses a biphasic life cycle to invade and replicate within its prey. Sometimes, predatory bacteria play an important role in natural ecosystem, affecting how nutrients are stored and released in the soil. The term microbiome encompasses all microorganisms associated with a specific organism or environment, including resident and transient microbiota. Resident microbiota are permanent inhabitants, while transient microbiota are temporary and can include pathogens. Hygiene and diet can influence these microbial populations. The diversity of the resident microbiota is significant, varying from location within the human body. For example, different bacterial species inhabit the mouth, with some preferring the tongue's surface while others thrive in cheek or gum areas. Variability exists not only between the individuals, but also within the different body sites of the same individual. The microbiota can change over time, with initial colonization occurring during natural birth. Lactobacillus species are among the first microbes to colonize in the newborn's gut, influencing long-term health. The microbiome continues to evolve as new microbes are introduced and others are eliminated. Resident microbiotas play a crucial role in human health by occupying niches that could otherwise be filled with pathogens. For instance, Lactobacillus species help to maintain vaginal acidity, inhibiting harmful yeasts. Disruptions such as antibiotic use can decrease these beneficial populations, potentially leading to infections and health issues. Assigning prokaryotes to specific species presents a significant challenge due to their asexual reproduction, which eliminates the possibility of classification based on interbreeding. Furthermore, prokaryotes lack distinct morphological features, making traditional classification methods such as shape, staining patterns, and biochemical differences insufficient. With advancement in technology, the analysis of nucleotide sequences in genes has emerged as a crucial criterion for microbial classification. In 1923, American microbiologist David Hendricks Burge published a Manual of Determinative Bacteriology, which summarizes the known types of bacteria using Latin binomial classification. This manual detailed the morphological, physiological, and biochemical properties of various bacteria and has undergone multiple updates to include newly discovered species. A more recent companion publication, the 5-volume Burge's Manual of Systemic Bacteriology, further expands on the original work, incorporating a broader array of species and providing updates, taxonomic description, and biological properties, including the officially recognized name of bacteria as outlined by the list of prokaryotic names with standing in nomenclature, or LPSN. Bacteria are traditionally classified based on their staining patterns, which are influenced by characteristics of their cell walls. This classification includes gram-positive, gram-negative, and atypical bacteria. Gram-positive bacteria have a thick peptidoglycan cell wall that retains the primary stain crystal violet during their recolorization process, resulting in a purple appearance after staining. Conversely, gram-negative bacteria have a thinner cell wall that allows the crystal violet to wash away, leaving them light red or pink after staining with the secondary counterstain, safranin. Atypical bacteria, which cannot be stained using the standard gram stain method, include species such as Mycoplasma, Chlamydia, and Rickettsia, and the latter being too small for evaluation by this procedure. Recent advancements in bacterial classification have led scientists to further divide gram-positive and gram-negative bacteria. This includes the identification of a special group of deeply branching bacteria recognized for their unique physiological, biochemical, and genetic traits. These deeply branching bacteria are considered to represent early evolutionary forms and thrive in extreme environments characterized by hot temperature, acidity, high ultraviolet light exposure, and anaerobic conditions. The classification of gram-negative bacteria has also refined into 3 main groups: Proteobacteria, Cryptophaga, Flavobacterium, Bacteroid (CFB), and Spirochetes. The Proteobacteria phylum encompasses a diverse range of gram-negative bacteria, some of which are significant human pathogens, including E. coli and Bordetella pertussis. The CFB group is notable, containing members of the normal human gut microbiota, such as bacteroids, which play an essential role in digestion. Spirochetes, characterized by their spiral shape, including pathogenic species like Treponema pallidum, which causes syphilis. Further details on these bacterial groups will be explored later in the chapter. In addition to their staining characteristics, gram-positive bacteria are also categorized based on their prevalence of guanine and cytosine nucleotides in their DNA. These are divided into low GC and high GC gram-positive bacteria. Low GC gram-positive bacteria contain less than 50% guanine and cytosine, and high include notable human pathogens responsible for diseases such as anthrax, tetanus, and listeriosis. High GC gram-positive bacteria possessing more than 50% of these nucleotides encompasses pathogens that cause diphtheria, tuberculosis, among other diseases, highlighting the importance of understanding bacterial classification for medical and scientific purposes. Bacterial phylogeny chart in the slide figure is based on the work of Hugenholz et al. Bacterial phyla are shown on the leaves, with the Firmicutes and Proteobacteria subdivided to the class level. Taxa are shown in italics. Their phylogeny is further broken down to the order level with the use of smaller branches. These 2 phyla account for roughly 3/4 of the described infectious bacterial species, with the noticeable exception of delta proteobacterial pathogens. Proteobacteria contains both important plant and animal pathogens. Proteobacterial plant pathogens are not clustered, but are observed in alpha, beta, gamma subdivisions. Some plant and animal pathogen species share same genus classification, Ralstonia and Pseudomonas, and for at least one species, Burkholderia cepacia. Different subspecies are plant and human pathogens. Of the 20 bacterial phyla currently recognized in NCBI taxonomy, 13 are not present in the figure above due to the absence of noteworthy pathogens. Some of these missing phyla are relatively important, like cyanobacteria, but most of the remaining phyla are restricted to a handful of species and/or environmental niches. Also of the interest is the relative weight of infectious agent within their respective phylum. While virtually all spirochetes and chlamydia constitute potential infectious agent due to their parasitic lifestyle, the phylum Actinobacteria has few pathogens related to the overall diversity of the phylum. It should be cautioned that our current view of bacterial diversity is biased towards cultivatable organism, which represent a smaller fraction of the bacterial diversity. Therefore, this compilation represents the well-organized infectious bacterial agents, but should in no way be considered exhaustive or complete. You may appreciate the complexity of the bacterial pathogen phylogeny in the previous slide. This slide presents the key for the symbols used in the previous slide chart. The symbols are broadly categorized on 1. Risk assessment, which is science-based, 2. Risk management, which is policy-based, and the number 3. Risk communication. The details are beyond the scope of this course, so the previous slide and this slide is for reference only. The learning objective focuses on describing the unique features of 5 classes within phylum Proteobacteria: alpha Proteobacteria, beta Proteobacteria, gamma Proteobacteria, delta Proteobacteria, and epsilon Proteobacteria, and providing examples of bacteria from each class. In 1987, microbiologist Carl Woese proposed that a diverse group of bacteria, initially termed purple bacteria, and their relatives should be classified as a separate phylum due to similarities in their genetic sequences. This group, known as Proteobacteria, encompasses both beneficial bacteria found in the human microbiota and various pathogens, highlighting its significance in microbiology. The alpha Proteobacteria is the first class of Proteobacteria characterized by their ability to thrive in oligotrophic environments, which are low in nutrients. These organisms can be found in various extreme habitats, including deep oceanic sediments, glacial ice, deep undersurface soil. Their unique adaptation allows them to survive in conditions where many other organisms cannot. Within alpha Proteobacteria, 2 notable groups are chlamydias and rickettsias, both of which are obligate intracellular pathogens. This means that a significant portion of their life cycle must occur within host cells, where they are metabolically inactive when outside. Chlamydia and rickettsia depend on their host cells for energy, and they are unable to synthesize their own adenosine triphosphate. Rickettsia species include several serious human pathogens, with R. rickettsii or Rickettsia rickettsii being particularly notable for causing Rocky Mountain spotted fever. This illness is a life-threatening form of meningoencephalitis, characterized by inflammation of the membranes surrounding the brain. Rickettsia rickettsii primarily infects by ticks and can be transmitted to humans through a bite of an infected tick, posing significant health risk in certain regions. Rickettsia prozovsky is a species spread by lice and cause epidemic typhus, a severe infectious disease often associated with warfare and mass migrations. This pathogen infects human endothelial cells, leading to inflammation of blood vessel linings. Symptoms include high fever, abdominal pain, in some cases delirium. A relative species, Rickettsia typhi, causes a milder form known as murine or endemic typhus, which remains prevalent in the southwestern United States during warmer months. Chlamydia represents another group within the alpha Proteobacteria. Members of this genus are gram-negative, obligate intracellular pathogens that possesses a remarkable ability to evade cellular defenses. They spread rapidly from host to host in their elementary body form, which is metabolically inactive but capable of entering epithelial cells where they become active and reproduce. The life cycle of chlamydia is crucial for understanding its pathogenicity and transmission. Chlamydia trachomatis, a notable human pathogen, is responsible for trachoma, an eye disease that can lead to blindness. Additionally, it causes lymphogranullar venereum, LGV, a sexually transmitted disease that can be mildly symptomatic, often presenting as a swollen lymph nodes or asymptomatic. This disease is highly contagious and is particularly common in settings such as college campuses, highlighting the importance of awareness and prevention in controlling its spread. Textbook Table 4.2. This is the second part of the textbook table 4.2. Beta proteobacteria are a class of bacteria characterized as eutrophs, meaning they require a high supply of organic nutrients compared to alpha proteobacteria, which thrive on minimal nutrients. These bacteria often inhabit areas of alternate between aerobic and anaerobic conditions such as intestine of mammals. Some genera within the class include species that are pathogenic to humans, leading to severe diseases. Notably, the genus Neisseria contains harmful species like Neisseria gonorrhoeae, responsible for gonorrhea, and Neisseria meningitidis, which causes bacterial meningitis. Neisseria are cocky bacteria that reside on mucosal surface in the human body and are known for their fastidious nature, making them challenging to culture. They require specific conditions, including high moisture levels, nutrient supplements, and carbon dioxide, and are microaerophilic, needing low oxygen levels for optimal growth. For identification purposes, Neisseria species are typically cultured on chocolate agar enriched with partially hemolyzed red blood cells. The heating of the blood agar partially hemolyzes the red blood cells, and the agar turns chocolate color as if the blood agar has been charged by heating. Their growth pattern is distinctive, often appearing as diplococci or pairs of cells resembling coffee beans. The bacterium Bordetella pertussis, a member of beta proteobacteria class within the order Burkholderillis, is the pathogen responsible for whooping cough or pertussis. It produces various toxins that paralyze the cilia in the human respiratory tract and directly damage respiratory tract cells, leading to severe cough. Textbook Table 4.3. Gamma proteobacteria is the most diverse class of gram-negative bacteria and includes several human pathogens, notably from the family Pseudomonaceae. A significant member of this family is the genus Pseudomonas, which includes the pathogenic species Pseudomonas aeruginosa. This bacterium is strictly aerobic, non-fermenting, and highly motile, often responsible for infections in wounds, burns, urinary tracts, and respiratory tissues, particularly in patients with cystic fibrosis or on mechanical ventilation. The treatment of P. aeruginosa infections is challenging due to its resistance to many antibiotics and its ability to form biofilms. Other notable Pseudomonas species include Pseudomonas fluorescens, known for its fluorescence, Pseudomonas putida, recognized for degrading xenobiotics. The family Pasteureliaceae contains several clinically significant genera, including pathogens affecting both humans and animals. Pasteurella hemolytica is known for causing severe pneumonia in sheep and goats, while Pasteurella maltosida can be transmitted from animals to humans via bites, leading to skin and tissue infections. The genus Haemophilus includes two important human pathogens, Haemophilus influenzae and Haemophilus ducreyi. Despite its name, Haemophilus influenzae does not cause the influenza, which is caused by the influenza virus, but can lead to upper and lower respiratory infections such as sinusitis and pneumonia. Historically, before vaccinations were developed, this bacterium was the major cause of invasive disease like meningitis in children. The order Vibrionalis is represented by the human pathogen Vibrio cholerae, a comma-shaped bacterium thriving in alkaline environment. It produces a toxin that triggers hypersecretion of electrolytes and water in the large intestine, resulting in severe watery diarrhea and dehydration. Other species in this order, such as Vibrio parahemolyticus and Vibrio vulnificus, are associated with gastrointestinal diseases and serious skin infections, respectively. Alivibrio fischeri, another member of Vibrionalis, has a symbiotic relationship with squid where it receives nutrients from the squid while providing bioluminescence that helps protect the squid from predators and also attract food in the night. The genus Legionella, part of the gamma proteobacteria, includes Legionella pneumophilia, the bacterium that causes Legionnaire's disease. This aquatic bacterium thrives in warm water environments, particularly in air conditioning units of large buildings. Legionella can spread through aerosols, leading to outbreaks contaminated water resources, often affecting residents within the same building. The name Legionella originates from the first known outbreak of the disease during the American Legion Convention in Philadelphia in 1976, as we discussed before. Enterobacteriaceae is a large family of enteric bacteria within the gamma proteobacterial class, known for its facultative anaerobic nature and the ability to ferment carbohydrates. This family is divided into two main categories: coliforms, which include Escherichia coli or E. coli, and can completely ferment lactose, and non-coliforms, which either cannot ferment lactose or do so incompletely. Notable pathogens within the non-coliform group include Salmonella species, Shigella species, and Yersinia pestis, which can cause various intestinal infection and diseases in humans. E. coli has been extensively studied since its discovery in 1886 by Theodor Escherichia. While many strains of E. coli coexist beneficially within humans, some produce Shiga toxin, a highly potent bacterial toxin that disrupts protein synthesis in target cells, leading to cellular death and severe gastrointestinal issues such as hemorrhagic colitis and potentially fatal hemolytic uremic syndrome. Other strains of E. coli are responsible for traveler's diarrhea, a common less severe illness that affects many travelers. The genus Salmonella, classified under non-coliform group, is complex, with an ongoing debate regarding the number of species it encompasses. Many previously classified species have been redefined as serotypes based on their surface molecules, reactivity with animal antisera. Certain serotypes of Salmonella are responsible for salmonellosis, which leads to inflammation of intestines, fever, vomiting, and diarrhea. While the serovar of S. enterica, serovar typhi, causes typhoid fever, presenting symptoms such as fever, abdominal pain, and skin rashes. This is the textbook table 4.4 and gamma proteobacteria, part 2. The delta proteobacteria is a small class of gram-negative proteobacteria that includes sulfate-reducing bacteria, or SRBs. These bacteria are named for their use of sulfate as the final electron acceptor in the electron transport chain. While few SRBs are known to be pathogenic, the species Desulfovibrio aureole is associated with periodontal disease, which affects the gums. Another significant group within the delta proteobacteria is the genus Delovibrio, which comprises parasites of other gram-negative bacteria. Delovibrio invades the host bacterium's cell, positioning itself in the periplasm, the space between the plasma membrane and the cell wall. This invasion allows Delovibrio to feed on the host's protein and polysaccharides, leading to lethal consequences for the infected cells. Additionally, Myxobacteria, another type of delta proteobacteria, thrive in soil environments, scavenging inorganic compounds. These bacteria are motile and highly social, engaging with other bacteria within and outside their group. They can form multicellular macroscopic structure known as fruiting bodies, which are currently the subject of ongoing research by biologists and bacterial ecologists. Myxobacteria also have capability of producing metabolically inactive spores called myxospores. This is the textbook table 4.5, is for the delta proteobacteria. Epsilon proteobacteria is the smallest class of proteobacteria, consisting of gram-negative microaerophilic bacteria that require minimal oxygen. Two significant genera within the class, Campylobacter and Helicobacter, both of which contain human pathogens. Campylobacter, particularly the species Campylobacter jejuni, is known to cause food poisoning characterized by severe enteritis, especially in developed countries where contaminated poultry products are a common source. Chicken are often carriers of Campylobacter jejuni, leading to contamination during meat processing. Helicobacter includes the notable bacterium Helicobacter pylori, which is a crucial part of the stomach microbiota, but also the primary cause of chronic gastritis and stomach and duodenal ulcers. Additionally, Helicobacter pylori has been associated with increased risk of stomach cancer. This bacterium is unique as it can thrive in the stomach's highly acidic environment by producing urease and other enzymes that help neutralize acidity, allowing it to survive and impact gastric health. Textbook Table 4.6 present the Epsilon proteobacteria. The learning objective focus on understanding non-proteobacteria, gram-negative bacteria, which include unique features and examples from three classes, spirochetes, the CFB group, and the Planktomycetes. Additionally, the objective emphasizes identifying and describing phototropic bacteria, which encompass both proteobacteria and non-proteobacteria, highlighting the diversity within these groups. Spirochetes are unique bacteria known for their long spiral-shaped bodies that can reach lengths up to 250 micrometer. Their thin structure possesses challenges for examination using traditional gram stain methods, leading to the preference for dark-field fluorescent microscopy. Spirochetes are highly motile organisms, utilizing an axial filament for movement. This filament, which is akin to a flagellum, is located within the spirochete's periplasmic space wrapped around the cell body, allowing for efficient propulsion. Among the genera of spirochetes, several are known to be human pathogens. The genus Treponema includes the notable species Treponema pallidum, which is further divided into four subspecies: Treponema pallidum pallidum, Treponema pallidum pertineo, the Treponema pallidum caratium, and Treponema pallidum endemicum. The subspecies Treponema pallidum pallidum is responsible for syphilis, a sexually transmitted infection that ranks as the third most common bacterial sexually transmitted infection in the United States, following by chlamydia and gonorrhea. The other subspecies are associated with tropical diseases affecting the skin, bones, and joints. Another significant genus of spirochetes is Borrelia, which includes several pathogenic species. Borrelia burgdorferi is the causative agent of Lyme disease, transmitted primarily through bites from certain tick species, such as Ixodes and Amblyomma. Lyme disease is characterized by symptoms such as distinctive bull's eye, rash, fever, fatigue, and in some cases severe arthritis. Additionally, Borrelia recurrentis is responsible for relaxing fever, underscoring the medical importance of these spirochetes. Flagellar structure. Spirochetes are a distinct phylum of motile bacteria characterized by unique cellular shapes, either helical or flat wave, depending on the species. A significant focus of research has been directed at understanding the motility and morphology of the spirochete species Borrelia burgdorferi, particularly due to its association with Lyme disease and its similarity to Treponema pallidum, the bacterium responsible for syphilis. Axial filament, also known as periplasmic flagella, are structurally akin to the flagella of other bacteria, consisting of a basal body motor complex, a hook, a filament. However, in spirochetes, these flagella reside in the periplasmic space, allowing them to function independently from the external environment. The motors at the base of the periplasmic flagella facilitate cellular motility, while the unique hook structure is formed by high molecular weight protein complexes that are stabilized through covalent cross-linking. The morphology of Borrelia burgdorferi is characterized by long aspect ratio with 7 to 11 periplasmic flagella attached at each end, creating a tightly wrapped ribbon that significantly aid in movement. Swimming of the bacteria with helical cell bodies. Treponema primatia has been periplasmic flagella aka axial filament, that rise from its two ends and run along the cell. Flagellar rotation drives circumferential motion of the loosely attached outer membrane, which generates torque, causing the helical cell to rotate about its axis and move forward. Leptospira eleni also has one short flagellum at each pole. The flagellum deforms the cell body into a helical shape at the front and a hook shape at the back. The helix propagates a backward moving wave, the hook gyrates, and the protoplasmic cylinder rolls the other way about the local helical axis to balance the torque. Borrelia burgdorferi has several flagellar motors at either end of the cell with filaments that overlap in the middle. The stiffness of the flagella and the protoplasmic cylinder are comparable. So the cell body undulates as well as rolls to generate propulsion. The figure A in the slide shows the Borrelia burgdorferi as a planar flat wave morphology that is the result of the periplasmic flagellum wrapping around the cell body between the protoplasmic cell cylinder and the outer membrane. The cross-section demonstrates that the periplasmic flagella are ordered in a ribbon-like manner, wrapping around the cell body. The PFs overlap in the center of the cell. Now, wild-type spirochetes are more resistant to compression under external stress applied by the atomic force microscopy imaging process. Recent studies utilizing a tapping mode of atomic force microscopy have demonstrated the essential role of periplasmic flagellas in maintaining the integrity of Borrelia burgdorferi cells under stress, which is the B in the figure of the slide. Sequential tapping mode AFM topography shows a wild type and B, a FLA-B mutant spirochete of Borrelia burgdorferi were taken with set points of say 0.8, 0.7, and 0.6. The lines actually indicated by various symbols in the images correspond to the height profiles indicated by the same symbol provided directly to the bottom of each image. Note that the axis limits vary between the different height profiles. The experiment used wild-type and mutant strains of Borrelia burgdorferi, specifically the FLA-B- and FLA-E- mutants, which were unable to form the flagellar filament and the hook, respectively. Although the motors of these mutants remain intact, their motility was greatly compromised, resulting in a rod-shaped morphology. The findings indicate that periplasmic flagella are vital for the bacterium's ability to resist cellular compression and preventing damage from external stresses, underscoring their importance in survival of the Borrelia burgdorferi. The gram-negative non-proteobacteria, including the genera Cytophaga, Fusobacterium, and Bacteroids, are grouped together as the CFB group. While these bacteria are phylogenetically diverse, they share similarities in their DNA nucleotide sequences. These rod-shaped bacteria thrive in anaerobic environments like the gums, gut, rumen of ruminating animals. They are efficient fermenters that can process cellulose, allowing ruminants to derive carbon and energy from their grazing activities. Cytophaga are unique motile bacteria that glide in aquatic environments, while fusobacteria are found in the human mouth and can lead to serious infections. The Bacteroids genus is the largest within the CFB group and comprises numerous species that are significant components of the human large intestine, accounting for approximately 30% of the gut microbiome. 1 gram of human feces can contain up to 100 billion Bacteroids cells. Most of these bacteria are mutualistic, providing benefits to humans by preventing harmful pathogens from establishing in the gut. However, a reduction in Bacteroids population, often due to antibiotic use, can create an environment that favors the growth of pathogenic bacteria and fungi, potentially resulting in secondary infections. Only some Bacteroids species are harmful. For instance, Bacteroid melaninogenicus may lead to infections in the individual with compromised immune systems. Planktomycetes are microorganisms found in water environments such as freshwater, saltwater, and brackish water. What makes Planktomycetes unique is their method of reproduction called budding. Instead of a single maternal cell dividing into two equal daughter cells through binary fission, the mother cell develops a bud that separates and becomes an independent cell. These buds are known as swarmer cells, which are motile and not attached to any surface. Shortly after, they will change into a sessile, immobile cell that develop a structure called the holdfast, enabling them to cling to the surface in the water. Only the sessile cells have the ability to reproduce. Textbook Table 4.7 Non-proteobacteria. Phototropic bacteria encompass a broad and diverse group of bacteria that utilize sunlight as their primary energy source. Rather than representing a specific taxon, this group includes both proteobacteria and non-proteobacteria. These bacteria synthesize ATP through photosynthesis, which can be categorized into two types, oxygenic and anoxygenic photosynthesis. While some cyanobacteria are known for oxygen production, the majority of the phototropic bacteria perform anoxygenic photosynthesis, which does not release oxygen. Among phototropic bacteria, there is a significant subset known as purple and green bacteria that utilize bacteriochlorophylls, pigments similar to plant chlorophyll, to perform photosynthesis. These pigments can manifest in various colors, including green, purple, or blue, and are often accompanied by carotenoids, which are red, orange pigments. The colors of these bacteria can vary greatly, allowing them to absorb light from different wavelengths. Traditionally, they are classified into sulfur and non-sulfur bacteria, with distinction made based on their pigmentation. Sulfur bacteria are known to carry out anoxygenic photosynthesis using sulfides such as electron donors, which result in the release of free elemental sulfur. In contrast, non-sulfur bacteria rely on organic substances like succinate and malate for their electron donation. Purple sulfur bacteria, characterized by their ability to oxidize hydrogen sulfide into elemental sulfur and sulfuric acid, gain their coloration from bacteriochlorophylls and carotenoids. The genus Chromatium, a model organism for bacterial photosynthesis research since 1950s, exemplifies purple sulfur gamma proteobacteria. These anaerobic microorganisms thrive in aquatic environments, utilizing carbon dioxide for growth, but require sulfides as electron donors for survival. Green sulfur bacteria, such as genus Chlorobium, utilize sulfide for oxidation, resulting in the production of significant amounts of green bacteriochlorophyll. These bacteria are of particular interest in the climate change discussions due to their ability to produce methane, a potent greenhouse gas. They employ at least 4 types of chlorophyll for photosynthesis, with bacteriochlorophyll being the most abundant, stored in specialized vesicle-like organelles known as chlorosomes. In contrast, purple non-sulfur bacteria, exemplified by the genus Rhodospirillum, use hydrogen for oxidation instead of hydrogen sulfide. These facultative anaerobes, which appear pink rather than purple, can metabolize nitrogen and are considered promising for biotechnological applications due to their potential to produce biological plastics and hydrogen fuel. Their metabolic versatility may contribute to sustainable practices in various industries. Green non-sulfur bacteria, like Chloroflexus, are similar to their sulfur counterparts but utilize different substrates for oxidation. Chloroflexus displays an orange hue when grown in the dark and turns green under sunlight. It also stores bacteriochlorophyll in chlorosomes and engages in anoxygenic photosynthesis, using low concentrations of organic sulfides or molecular hydrogen as electron donors. This bacterium can survive in the dark if oxygen is present and demonstrate gliding motility rather than flagella. Additionally, Chloroflexus thrives at a wide range of temperature, indicating its thermophilic nature. Cyanobacteria, a large and diverse group of phototropic bacteria, are known for their blue-green color, which comes from the chlorophyll in their cells. These organisms perform oxygenic photosynthesis, producing significant amounts of gaseous oxygen. Scientists believe that cyanobacteria were instrumental in transforming the Earth's atmosphere from an anoxic state to the oxygen-rich environment we experience today. A process occurred 1 to 2 billion years ago. These bacteria are remarkably adaptable, thriving in a variety of habitats such as marine and freshwater environments, soil, and even rocky surfaces. Their resilience allows them to survive in extreme temperature conditions, including frigid environments of Antarctic. Cyanobacteria can exist as unicellular organisms or colonies that can take filamentous forms, creating sheaths or biofilms. A notable feature of many cyanobacteria is their ability to fix nitrogen, a process that converts molecular nitrogen into useful compounds like nitrites and nitrates, facilitated by specialized cells called heterocysts. In terms of photosynthesis, cyanobacteria utilizes oxygenic process employing chlorophyll A, which is also found in plants and algae. They possess additional pigments, phycocyanin and cyanophycin, that contributes to their distinctive blue coloration. These pigments are housed in a specialized organelles known as phycobilisomes and in thylakoids, which share similarities with photosynthetic structure found in plants. This has led scientists to hypothesize that modern plants may have evolved from the endosymbiosis of ancestral eukaryotic cells with photosynthetic bacteria. However, cyanobacteria can also pose risks to human health. Certain genera, such as Microcystis, can create harmful blooms in water bodies, resulting in dense mats that produce toxins. These toxins have been linked to several health issues, including liver tumors and nervous system disease in both animals and humans. Thus, while cyanobacteria play a crucial role in ecosystem and possesses fascinating biological properties, their potential negative impacts cannot be overlooked. Textbook Table 4.8, Phototropic Bacteria. The link will take you to an HHMI biointeractive module featuring a virtual Winogradsky column, allowing exploration of microbial diversity, metabolic strategies, and geochemical gradients within sediments. This virtual tool serves as an alternative to hands-on labs, enabling students to learn about the process in both virtual and actual Winogradsky column. The learning objectives focus on understanding the distinctive characteristic of high GC and low GC gram-positive bacteria, highlighting the similarities and differences between these two bacterial groups and providing examples of a bacterium from each category commonly associated with high GC and low GC groups. Prokaryotes are classified as gram-positive when they possess a multi-layered peptidoglycan matrix in their cell walls, which allow them to retain the primary stain crystal violet during the gram stain procedure. As a result, these organisms appear purple under the bright field microscope. Historically, the ability to retain gram stain has been a key criteria for classifying prokaryotes, although some do not respond well to the staining process. Recent advances in nucleic acid biochemistry have been introduced new classification methods for gram-positive prokaryotes, focusing the guanine to cytosine ratios in DNA and the composition of 16S ribosomal RNA subunits. Microbiologists now recognize two main groups of gram-positive bacteria: actinobacteria, which are high GC bacteria with over 50% guanine and cytosine nucleotides in their DNA, and bacilli, which are low GC bacteria with less than 50% guanine and cytosine nucleotides in their DNA. Actinobacteria, derived from the Greek word rays and small rods, are a diverse group of bacteria with varying microscopic appearances ranging from thin filaments to rods to coccobacilli. This group includes some of the largest and most complex bacteria, as well as some of the smallest independent organisms. While most actinobacteria thrive in soil, some are found in aquatic environment, with majority being aerobic. A notable characteristic of actinobacteria is their cell wall, which contains multiple types of peptidoglycans. The genus Actinomyces represent a well-studied subgroup of actinobacteria, playing a significant role in soil ecology. Some actinomyces species are opportunistic pathogens found in the human mouth, leading to infections such as periodontitis and oral abscesses. Noteworthy is a species A. israeli, an anaerobic bacterium responsible for endocarditis and inflammation of the heart's inner lining. Another important genus is Mycobacterium, which features bacilli covered with a protective mycolic acid coat. This waxy layer shields the bacteria from certain antibiotics, prevents desiccation, and hinders gram stain penetration, necessitating a special acid-fast staining technique for visualization. Mycobacterium is known for causing various infectious diseases, including Mycobacterium tuberculosis, the agent of tuberculosis, which predominantly affects the lungs. With an estimated one-third of the global population infected and millions of new cases annually, treating Mycobacterium tuberculosis is complex, often requiring prolonged combination of medications due to the rise of multi-drug resistant strains. Mycobacterium lepori, another pathogenic Mycobacterium species, is responsible for the Hansen's disease or leprosy. This chronic condition affects peripheral nerves and the skin, leading to loss of pain sensation and making individuals more vulnerable to secondary infections and injuries due to skin lesions and respiratory tract complications. Corynebacterium, another genus of actinobacteria, is characterized by the presence of diaminopimelic acid in their cell walls and often forms palisades or V-shaped arrangement of rod-shaped cells. Many Corynebacterium species are non-pathogenic. However, Corynebacterium diphtheria is a significant exception as it causes diphtheria, a potentially fatal disease, particularly in children. The bacterium produces a toxin that leads to the formation of pseudomembrane in the throat, resulting in severe symptoms if left untreated. Lastly, the genus Gardnerella includes the species Gardnerella vaginalis, which is classified as gram variable due to the inconsistent results during gram staining. Based on genomic analysis, it is characterized within the high GC gram-positive group. G. vaginalis is known to cause bacterial vaginosis in women, which can present with mild or undetectable symptoms but may lead to complications during pregnancy if not addressed. Textbook Table 4.9, Part 1, Actinobacteria, high GC gram-positive. Textbook Table 4.9, Part 2, Actinobacteria, high GC gram-positive. Low GC gram-positive bacteria contain under 50% guanine and cytosine in their DNA, and this category includes several genera of pathogenic bacteria. Clostridia is a large and diverse class of low GC gram-positive bacteria, with Clostridium being the most studied genus. These rod-shaped obligate anaerobes produce endospores, allowing them to endure in anaerobic environments such as nutrient-rich soil and aquatic sediments, where their endospores can survive for many years. Clostridium species are known to produce a diverse range of protein toxins, with several species identified as human pathogens. Among this, Clostridium perfringens is the third most common cause of food poisoning in the United States and is also responsible for severe condition known as gas gangrene. These occurs when Clostridium perfringens endospores enter a wound, germinate, and produce toxin that lead to tissue necrosis. Other notable species including Clostridium tetani, which causes tetanus through a neurotoxin that disrupts nerve impulse inhibition, resulting in potentially fatal spastic paralysis. Additionally, Clostridium botulinum is infamous for producing the botulinum neurotoxin, recognized as the most lethal biological toxin found on Earth, which can cause flaccid paralysis by blocking acetylcholine release at neuromuscular junctions. Although botulinum toxin can lead to fatal botulism in rare instances, it is also utilized in small doses for treating muscle conditions and in cosmetic procedures. Clostridium difficile, more commonly known as a C. diff, contributes to hospital-acquired infections, often causing serious colitis, particularly in immunosuppressed patients or those undergoing antibiotic treatments that disrupt their gastrointestinal microbiota. The order Lactobacillus includes the low GC gram-positive bacteria featuring both rod-shaped and round forms in the genera Lactobacillus, Leuconostoc, Enterococcus, and Streptococcus. Bacteria from the latter three genera are generally spherical or oval, frequently arrange themselves in chains. Streptococcus, derived from the Greek term twisted chain, is a genus responsible for various infectious diseases in humans. These bacteria, commonly called streptococci, are categorized using serotypes known as Lancefield groups, as well as their capacity to lyse red blood cell when cultured on blood agar. Among these species, Streptococcus pyogenes is classified as Lancefield group A and is identified as beta hemolytic streptococcus. This particular species is recognized as a pyogenic pathogen due to the pus formation associated with its infection, making it a significant health concern. S. pyogenes is a leading cause of bacterial pharyngitis, commonly known as strep throat. In addition to the throat infection, it is responsible for various skin infections, range from mild conditions like impetigo to severe life-threatening diseases such as necrotizing fasciitis, often referred to as a flesh-eating disease. The potential severity of these infections highlights the importance of understanding and addressing S. pyogenes in clinical settings. In contrast, non-pyogenic streptococci comprise a group of species that are not classified under any Lancefield group but are primarily found in the human mouth. These bacteria are mostly commensal, meaning they coexist without causing harm. However, some, such as S. mutans, are linked to development of dental caries, indicating that even non-pyogenic streptococci can have clinical implications. Another significant species, S. pneumoniae, is not classified under any Lancefield group. This bacteria is typically observed as diplococci or pairs of cells, rather than characteristic long chains of most streptococci. Known since the 19th century for causing pneumonia and respiratory infection, S. pneumoniae is capable of leading to a wide variety of other diseases including meningitis, septicemia, osteomyelitis, endocarditis, particularly affecting vulnerable populations such as newborns, the elderly, and the immunocompromised individuals. The class Bacilli includes a diverse array of bacteria that are primarily bacillus-shaped but also encompasses coccus-shaped genera. Notably, two clinically significant genera within this class are Bacillus and Staphylococcus. The morphogenic diversity of bacilli suggests a broader range of characteristics and function than implied by the name alone. Genus Bacillus characteristic. Bacteria from the genus Bacillus are typically bacillus-shaped and are capable of producing endospores. They can be classified as aerobes or facultative anaerobes, which allow them to thrive in various environments. Several bacterial species find an application in industries, contributing to the production of antibiotics like Barnase, enzymes such as alpha amylase, BAM1H1 restriction endonuclease, and detergents including subtilisin. Pathogenic species of Bacillus. Among the Bacillus species, two notable pathogens are Bacillus anthracis and Bacillus cereus. Bacillus anthracis is responsible for anthrax, a severe disease that can affect both animals and humans, leading to symptoms such as charcoal black ulcers, severe enterocolitis, pneumonia, and brain damage. If left untreated, anthrax can be fatal. Bacillus cereus is associated with food poisoning and is characterized by its rod-shaped appearance and milky white colonies on blood agar. Bacillus thuringiensis also have significant uses. Another significant species, Bacillus thuringiensis, which produces substances that serve as insecticides due to their toxicity to insects. This bacterium plays a crucial role in agricultural pest control, highlighting the beneficial application of certain Bacillus species beyond their pathogenic potential. The genus Staphylococcus, while part of the class Bacilli, is distinct in that its bacteria are coccus-shaped. The name Staphylococcus derives from the Greek word bunches of grapes, reflecting their appearance under a microscope. These bacteria are facultative anaerobic, halophilic, and non-motile. The two most well-studied species are Staphylococcus epidermidis and Staphylococcus aureus, each with important implications for human health. Staphylococcus epidermidis primarily inhabits human skin and is generally considered non-pathogenic in individuals with healthy immune systems. However, in immunocompromised patients, it can lead to infections, particularly in skin wounds and prosthetic devices such as artificial joints and heart valves. In hospital setting, Staphylococcus epidermidis poses a risk, especially for patients with compromised immune systems due to association with intravenous catheter infections. Staphylococcus aureus is responsible for a wide range of infections, including skin conditions like boils and cellulitis. Certain strains can produce enterotoxin, leading to staph food poisoning, while others are linked to toxic shock syndrome, which is life-threatening. Alarmingly, many strains of Staphylococcus aureus have developed antibiotic resistance, including methicillin-resistant Staphylococcus aureus, or MRSA, and vancomycin-resistant Staphylococcus aureus, or VRSA. These strains present significant treatment challenges and can be contagious, posing serious risks in healthcare environments where vulnerable populations are present. Mycoplasma species are unique because they lack a cell wall, which means they do not respond to gram staining. Despite these, they are categorized among the low GC gram-positive bacteria. The Mycoplasma genus comprises over 100 species, all sharing distinct traits. These cells are very small, with some measuring about 0.2 micrometer in diameter, making them smaller than certain large viruses. Their absence of a cell wall allows for pleomorphism, enabling them to adopt various shapes resembling tiny animal cells. This variability in form can complicate their identification. One notable species, M. pneumoniae, is responsible for mild type of pneumonia known as walking pneumonia or atypical pneumonia, which is generally less severe than other bacterial or viral pneumonia types. The figure A shows a 3D WC model of Mycoplasma genitalium visualized with CellPack GPU. Two clipping planes progressively hide parts of the model. The upper section highlights the ribosome, magenta, DNA yellow, and mRNA pink filaments. The central section shows the bacterial nucleoid in the context of its soluble macromolecules, DNA-binding proteins in orange, cytoplasmic proteins in shades of blue, tRNAs in bright pink. The lower section actually shows the cell membrane, gray-green, with associated membrane proteins with shades of green. Section 4.5, Deeply Branching Bacteria. Deeply branching bacteria are the oldest known life forms on Earth and are thought to be the closest relatives of the last universal common ancestor, LUCA. They're important for understanding the evolution of life. The phylogenetic tree illustrates the evolutionary relationship of organisms, with the trunk symbolizing a common ancestor known as the last universal common ancestors, LUCA. Deeply branching bacteria such as the genus Acetothermus are considered some of the earliest life forms, dating back approximately 3.5 billion years. These organisms branch from the LUCA, indicating their significance in understanding the origins of life on Earth due to their close evolutionary ties to the ancient ancestor. Research suggests that deeply branching bacteria might reveal insights into the structure and function of ancient life forms. It is hypothesized that these early bacteria were like thermophiles and hyperthermophiles, flourishing in extremely high temperature environments. Acetothermus paucivorans, a notable example discovered in 1988, thrives optimally at 58 degrees Celsius and is recognized as the closest living relative to LUCA, emphasizing its evolutionary importance. The class Aquificae contains deeply branching bacteria adapted to extreme conditions similar to those present on early Earth. Genus Equifax includes hyperthermophile that inhabit environments like hot springs and underwater volcanoes. For instance, A. pyrophilus can endure temperatures exceeding 138 degrees Celsius and utilize inorganic material for nutrients. These bacteria also exhibit strong resistance to ultraviolet light and radiation, supporting the theory that their ancestor evolved during a time when Earth was inhospitable and full of radiation. Thermatogae, another class of deeply branching bacteria, consist mainly of hyperthermophilic and some mesophilic species. They are characterized by a unique sheath-like outer membrane, or toga, and a distinct cell wall structure. Genera like Thermatoga include species that thrive in high-temperature environments such as T. maritima near thermal ocean vents, showing their adaptability and potential for industrial application through hydrogen production. Deinococcus radiurans is a remarkable bacterium known for its extreme resilience to harsh conditions, making it one of the most radiation-resistant organisms identified. These extremophile can endure cold temperatures, dehydration, vacuum condition, and acidic environments, earning it the title of polyextremophile. In January 1998, it was recognized by the Guinness Book of World Records as the world's most radiation-resistant bacterium or life form. However, it is important to note that the Archaea thermococcus gamma tolerans is actually the most resistant organism to radiation. The discovery of D. radiurans occurred in 1956 when Arthur Anderson was conducting experiments at the Oregon Agricultural Experiment Station in Corvallis, Oregon. The research aimed to establish whether the canned food could be sterilized using high dose of gamma radiation. During the experiments, a tin of meat was subjected to a radiation dose believed to eliminate all known life forms. Surprisingly, the meat spoiled, leading to the isolation of D. radiurans. The name Deinococcus radiurans originates from the ancient Greek word dinios and kokos, which means terrible grain berry, and from the Latin words radius or durare means radiation surviving. Previously, it was known as Micrococcus radiurans. Due to its extraordinary hardness, it has been affectionately named as Conan the Bacterium, referring to the fictional character Conan the Barbarian. Initially classified in the genus Micrococcus, further analysis of ribosomal RNA sequences and other evidences led to the reclassification into its own genus Deinococcus, which shares a close relationship with the genus Thermus. In addition, Deinococcus radiurans is used for studying cell cycle in bacteria. The right side figure is hyperlinked to an online movie showing the cell cycle in 6 phases. When cultured and exposed to ionizing radiation in liquid media, Deinococcus radiurans can survive doses up to 25 kg, but research by Horn in 2022 indicates that desiccated and frozen cells can withstand significantly higher radiation level up to 140 kg, which translates to a theoretical survival time of 280 million years at a depth of 10 meters beneath the Martian surface. This survival duration is relatively short when compared to the 2 to 2.5 billion years since liquid water vanished on Mars, making it unlikely for microbes to survive at a depth accessible to rovers equipped with drilling systems. Horn et al. Also proposes that meteoroid impacts may have sent Martian soil into a subsurface and temporarily heated the environment, possibly allowing extremophiles similar to D. radiurans to emerge briefly before succumbing to freezing conditions again. Therefore, for future missions such as European Rosalind Franklin rover targeting younger impact craters could enhance the likelihood of finding dormant extremophile microorganisms on the Martian surface, which are shielded from the harmful ionizing radiation. Note, kilogray is a unit used to measure the amount of energy absorbed by a material during irradiation. It is the international unit of absorbed radiation. The learning objective focus on understanding the distinctive characteristic of different categories of archaea, exploring the reasons why archaea may not linked to human microbiomes or diseases, and identifying common examples of archaea found in specific environmental habitats. The slide figure shows a comparison of bacterial and archaeal cells. Organisms in the domain archaea, like those in the domain bacteria, are unicellular, but they exhibit notable structural differences. The archaeal cell membrane is characterized by other linkages with branched isoprene chains, contrasting with ester linkages and unbranched fatty acids found in bacterial membranes. Additionally, while archaeal cells walls do not contain peptidoglycan, some possesses a similar substance known as pseudopeptidoglycan and pseudomurine. Furthermore, the genomes of archaea are larger and more complex compared to those of bacteria. Domain archaea exhibits a remarkable diversity akin to that of domain bacteria, with representatives thriving in various habitats. Many archaea are mesophiles, while a significant number are extremophiles, thriving in extreme conditions such as high temperature, extreme salinity, or other environments that are inhospitable to most life forms. The unique metabolic process, including methanogenesis, allows them to adapt to these harsh environmental surroundings, a capability not found in bacteria or eukaryotes. Classifying archaea is challenging due to the size and complexity of their genomes. Taxonomists generally categorize archaea into 5 major phyla: Crinarchaeota, Euryarchaeota, Coraarchaeota, Nanoarchaeota, and Thamarchaeota. However, it is believed that many other archaeal groups remain unstudied and unclassified, indicating a vast diversity yet to be explored within this domain. Despite their diversity, archaea are largely absent from the human microbiota, and none have been identified as causing infectious disease in humans, animals, plants, or other microorganisms. Nevertheless, many archaea play a crucial role in environmental processes, which may indirectly influence human health and ecosystems. Their contributions to the biogeochemical cycles and other ecological functions highlight their importance despite the lack of direct association with human and health issues. Crinarchaeota is a diverse class of archaea that encompasses a wide range of genera and species, varying greatly in morphology and growth requirements. These organisms are exclusively aquatic and are believed to be among the most abundant microorganisms in the oceans. While many Crinarchaeota are hyperthermophiles, capable of thriving at extreme temperatures, some of the genus, as is Pyrolobus, can grow at temperatures reaching up to 130 degrees Celsius. This remarkable adaptability allows them to inhabit some of the most challenging environments on Earth. Among significant genera with the Crinarchaeota is Sulfolobus, which consists of thermophiles that thrive in temperatures around 70 to 80 degrees Celsius and prefer acidic condition with pH 2 and 3. Sulfolobus can survive in both aerobic and anaerobic environments, utilizing metabolic process similar to heterotrophs when oxygen is present. In anaerobic setting, they oxidize sulfur, resulting in the production of sulfuric acid, which is stored in granules. These organisms have applications in biotechnology, particularly for producing thermostable and acid-resistant proteins known as effitins, which can bind and neutralize various antigens that incite human responses. Another notable genus of Thermoproteus, characterized by strictly anaerobic organism with an optimal growth temperature of 85 degrees Celsius. Thermoproteus species are motile due to their flagella and possesses a unique cellular membrane with a monolayer of lipid structure, differing from the typical bilayer of archaea or the bacteria. Their autotrophic metabolism involves the reduction of sulfur or molecular hydrogen, using carbon dioxide or carbon monoxide for carbon sources. As one of the earliest branching genera of archaea, Thermoproteus serves as a living representation of some of the Earth's most ancient life forms, highlighting the evolutionary significance of this class. The phylum Euryarchaeota encompasses various distinct classes, particularly the methanogens found in the class Methanobacteria, Methanococci, Methanomicrobia. These unique archaea have the ability to reduce carbon dioxide in the presence of hydrogen, resulting in the production of methane. Methanogens are known for their resilience, thriving in extreme environments and reproducing in a wide range of temperatures from below freezing to boiling. Their presence has been documented in diverse locations, including hot springs and beneath ice in Greenland. Some researchers even speculate that similar organisms could exist on Mars due to resemblance between the gases produced by methanogens and those found in Martian atmosphere. Methanogens play a significant role in creation of anoxic sediments by generating hydrogen sulfide, which contributes in the formation of marsh gas. Additionally, they produce gases in the digestive system of ruminants and humans. Notably, certain genera of methanogens, such as Methanosarcina, can metabolize and produce methane even in the presence of oxygen, although most are classified as strict anaerobes that thrive without oxygen. Another important class within the phylum Euryarchaeota is halobacteria, which includes halophilic archaea that thrive in high salinity environments. These organisms require sodium chloride concentration close to saturation, around 36%, and can be found in locations such as Dead Sea and some saline lakes in Antarctica and Central Asia. The remarkable characteristic of halobacteria is their ability to perform photosynthesis using protein bacteriorhodopsin, which imparts a distinctive purple hue to both of the organisms and their habitats. Among the notable species of halobacteria is the Halobacterium salinarium, believed to be one of the oldest living organisms, with DNA isolated from fossils dating back to 250 million years. Another significant species, Halopharax volcani, exhibits an advanced ion exchange system that allows it to maintain salt concentration balance in high temperature environments. These adaptations highlight the remarkable diversity and resilience of archaea within extreme conditions. Here is a microbiology connections, finding a link between archaea and a disease. Archaea are unique microorganisms that are not known to cause disease in humans, animals, plants, or even other archaea. While many archaea are extremophiles living in extreme environments, some genera and species are mesophiles and can inhabit human-animal microbiome, though this is rare. Notably, some methanogens are found in the human gastrointestinal tract, but there is currently no reliable evidence linking archaea to any human diseases. Despite this, researchers have explored potential connections between archaea and human health issues. In 2004, a study by Lepp et al. Indicated that the archaean Methanobrevibacter oralis is present in the gums of patients with periodontal disease, suggesting a possible link to the condition. However, further investigation revealed no causal relationship. Instead, it appears that periodontal disease creates an anaerobic condition in the mouth that M. oralis can then inhabit. Another microbiology connection is the Human Microbiome Project. The Human Microbiome Project was initiated by the National Institute of Health in 2008 with the primary aim of creating a comprehensive repository of gene sequences from significant microbes associated with humans. This project is designed to assist biologists and clinicians in understanding the complex dynamics of the human microbiome and its relationship with various diseases. A collaborative network of laboratories has been collecting data through swabs taken from different areas of the skin, gut, mouth, and hundreds of individuals. One of the significant challenges in studying the human microbiome is the difficulty in culturing many of the microbes that reside in the human body. Research indicates that only about 1% of the bacteria in nature can be cultured, leaving 99% unrepresentable in traditional laboratory settings. To overcome this limitation, scientists have turned to metagenomic analysis, which examines genetic material extracted directly from the microbial communities instead of relying solely on cultured species. This innovative approach enables researchers to analyze the genetic material of all microbes present in the microbiome, providing a more comprehensive understanding of these communities. A notable accomplishment of the Human Microbiome Project is the establishment of the first reference database for microbiomes inhabiting the human body. While many of these microbes are beneficial, the presence of pathogenic bacteria has also been discovered. For instance, the conjunctiva of the eye contains 24 genera of bacteria, including several pathogenic species. Similarly, the mouth hosts various species of the genus Streptococcus, some of which are known to be pathogenic. This finding prompts question about why certain microorganisms can coexist harmlessly in some individuals while posing serious health risk to others. Additionally, the project was uncovered numerous previously uncultured organisms, such as 174 new bacterial species identified in studies of the gut microbiota. Looking ahead, one of the project's goals is to characterize the human microbiota in individuals with various diseases to explore potential links between microbiota composition and disease susceptibility. By analyzing the microbiome of patients suffering from specific conditions, researchers hope to uncover new strategies for combating diseases, thereby enhancing our understanding of the microbiome's role in health and illness. The first and the second phases of the NIH Microbiome Project. The slide figure shows a 10-year NIH Microbiome Project, HMP, comprised two phases, HMP1 and HMP2, aimed at creating resources for the research community, including reference sequences, multi-omic datasets, and analytical tools. HMP1 focuses on characterizing microbial communities across various body sites in healthy adults and included demonstration projects targeting specific diseases. In contrast, HMP2 analyzed the biological properties of the host and microbiome through longitudinal studies on pregnancy, inflammatory bowel disease, and prediabetes, utilizing multi-omic analysis to track changes over time in microbial composition and host characteristic. All generated sequences, data, and tools from both phases are available through the HMP Data Collection Center, DCC, and other repositories, providing a central resource for the researchers. Here is one clinical focus with a resolution. Marcia, a 20-year-old university student, recently returned to the United States after interning as a medical assistant in Nigeria, where she worked to enhance access to tuberculosis testing. Upon her return, she initially attributed her fatigue to a jet lag, but her symptoms persisted and worsened. Along with fatigue, Marcia experienced coughing, night sweats, loss of appetite, and low-grade fever, 37.4 degrees Celsius, about 99. After 2 weeks, her conditions deteriorated, leading to her to cough up blood sputum with white clumps. Concerned, Marcia scheduled a doctor's appointment to address her escalating symptoms. During her visit to the physician, an examination revealed abnormal lung sounds, prompting the doctor to order a chest radiograph and a sputum sample for further analysis. The radiographic results showed cavities and opacities in her lungs, indicating potential underlying diseases. Given Marcia's recent travel to Nigeria and her exposure to tuberculosis during her internship, her physician suspected tuberculosis as the likely cause of her symptoms and radiograph findings. To manage her condition, Marcia was advised to remain at home, wear a respiratory mask, and isolate herself to prevent the spread of infection. She was also recommended to take a semester off from school and was prescribed a regime of antibiotics including isoniazid and rifampin to treat her tuberculosis over the next 3 months. Additionally, doctors may sometimes prescribe a third antibiotic, ethambutol. The bacteria suspected causing TB are known to be highly resistant to antibiotics because of thick, waxy mycolic acid in their cell wall, leading to the rapid resistant development. To counteract these effects, a combination antibiotic therapy is employed. The treatment plan was designed to facilitate her recovery while reducing the risk of transmitting the disease to others. Subsequent laboratory tests confirmed the presence of acid-fast bacteria in Marcia's sputum, consistent with the Mycobacterium tuberculosis. Although the initial cultures showed slow growth, distinctive colonies were identified after 4 weeks, leading to a definitive diagnosis. Following 9 months of consistent treatment, Marcia successfully recovered from her tuberculosis infection, highlighting the importance of timely medical intervention and appropriate care for infectious diseases. Here are some links to learning and educators' resources. And thank you.